Hi, this is Miss Litton, and this is 7th period support. Did you get your haircut? Looks so cute in a non-creepy teacher way. Okay, this is chapter 24 for Honors Bio. Say hi.
Hi.
And when we look at all the things that we talked about in this particular chapter, the first thing we talked about was just straight up DNA, and we looked at the history of it. And we had some key experiments. Do you remember all the different scientists we studied? Can you think of some? Hershey and Chase, Watson and Crick. Who else? What? Rosalind Franklin. Who else? Chargaff. Good. Okay. We also then talked about the structure of DNA, right? Because a lot of that work was done also, that research in order to come up with the structure of DNA. Who got the Nobel Prize for it? Watson and Crick. Okay. And you would say most simply that DNA, what shape is it?
Helix.
Not just a helix, a double helix. Good. Okay. Then the next thing we talked about is that if once we know the structure of DNA, we wanted to look at how does it replicate? How do we make more DNA? And that was something that would occur during the S stage of interphase, right? So that's one whole big chunk. DNA, its history, its structure, and how it replicates. Okay? Then we talked about, ultimately, we say, if we're going to describe DNA, it codes for all of our traits, right? If this is our genotype, then our traits would be our what? Phenotype. So we wanted to understand, how does your DNA code for your traits? So then the next thing we talked about is how we can convert this DNA to what? RNA, right? And that process is called, do you remember? Transcription. Transcription is when you go from DNA to RNA. We also know that there are 3 kinds of RNA. What are they?
mRNA.
M, T, and what? R. RNA. Okay? mRNA, tRNA, and rRNA. And they all have different purposes, yes? Okay, then the third step is we use that RNA to build a protein. Okay, and that process is called translation. Okay, that's the basic premise where all the details that I give you today have to fall underneath this umbrella in one of these places. Okay, this is what you for sure need to know. You need to know the term transcription and translation. And in translation, you're using the RNA to build a protein, right? And to have any RNA, you have to do transcription. DNA, DNA making more DNA is called replication. Okay, just making more of it and the copy of that. All right, so let's start with the first part, and that was the history of DNA. So one thing, remember, we were talking about is they knew inside cells that it contained both protein and DNA, and it kind of seemed like would make more sense if protein, because protein has how many different components to protein? 20, because there's 20 amino acids, right? But when you look at nucleotides that build up DNA, there are only 4. So it just made more sense if our genetic code was a protein. So then we had the work that was done by Hershey and Chase, because we always chase down a Hershey bar, so we're chasing down what is our hereditary material. So then they looked at a virus, and they labeled the protein parts of the virus, and they labeled the nucleic acid part of the virus, Hershey and Chase. So the nucleic acid part, remember, nucleic acids are built out of nucleotides. Nucleotides consist of 3 things. What are they? Base, sugar, phosphate. So the phosphorus was on the phosphate. That's what you want to remember. Okay, the sulfur was on the protein. And when they analyzed what got injected, they found the radioactive isotope inside the bacterium. So they knew that was the hereditary material, not the phosphorus. Okay, then we had some other experiments as well. One of those was Chargaff. And I don't know why I fixed this one, but this is the yucky one coming up, so I apologize. The yucky version, the bad version. Chargaff said a couple of things. One thing that Chargaff said was the composition of DNA is going to vary. You know, how many adenines, thymines, guanines, and cytosines in each species you have. But here's what's for sure. However many adenines you have, that will be equal to the number of thymines. However many guanines, that will be equal to the number of cytosines. Perfect. So that was good information. Okay. Then you talked about Rosalind Franklin. And so what she was doing was X-ray diffraction studies. So she was sending X-rays through crystallized DNA and getting a pattern. And what she could see in that pattern was that there was some sort of helical nature to DNA. That there were repeating units and it was the same diameter. Okay, so that information coupled with Chargaff is what helped Watson and Crick, who got the Nobel Prize, to come up with the double helix. Because Chargaff's data would explain why the C's and G's are paired and the A's and T's are paired. Okay, and Rosalind Franklin and Maurice Wilkins' work would explain the diameter is the same because you always have a purine connected with a pyrimidine. So it's not like you get two purines, that would be like 4 rings across as opposed to always 3 rings, right? Because you always have a purine that has 2 rings with a pyrimidine that has 1 ring. And it's definitely helical in structure. The diameter stays the same, and there are these repeating units. So that was history of DNA, and I've given you structure, a little bit more detail on the structure. We know that a nucleotide consists of a base, a sugar, and a phosphate group. We know that, okay? A base, a sugar, and a phosphate. But we also learned some things about the sugars. When we look at a sugar, where the nitrogenous base connects with the sugar, so wherever your nitrogenous, it's going to drive me crazy that we have the wrong notebook. I'm sorry, it's my fault. Where the nitrogenous base is connected to the sugar, that is always carbon number 1. And if you want to climb up on the roof, you can't because there's not another carbon up there. What's up there instead? Oxygen. So we come down, here's 2', 3', 4', and the chimney makes 5'. Now the reason why this becomes important is when you look at the structure of DNA, you can look right here. Mrna. Okay? We're going to look right here, and we're going to see the nitrogenous base coming in, and where it touches the sugar, that's carbon 1. We can't go on the roof, because what's up on the roof? Oxygen. So we come down to 3. That's why this is referred to the 3' end, because that's the first carbon edge that you can see, is that 3' end. Now we know DNA is parallel, but it's anti-parallel. They run 2 different directions. You can see all these houses look like they're right side up. Here is 1, 2, 3, 4. Chimney makes 5' end. When we go to the other side, it looks like the chimneys the chimneys and the houses are upside down. Okay, so this is the 5' end. Yes?
In this diagram, which one would be the leading strand and which one would be
It would depend where you started. If I was starting right here, if this is where I was starting, here's my eyeball, and I'm starting right here, this would be the leading, and this would be the lagging. But if I was making a mess on the if I was starting up here, then this would be the leading strand over here on the right, and that would be the lagging. It's wherever the 3' end is. Yeah. Because that's why we call it DNA polymerase 3. Because that's a 3' end. Does that make sense? Okay. So, and that's why I wanted to teach you about the sugar, so the leading and lagging would make more sense. If you look here, right, we can see them all numbered that way. So if we were starting from the bottom, would we go bottom left or bottom right? Which one would be the leading?
bottom left?
Bottom left. And if we were coming from the top, it would be top.
Right.
If we were coming from up top, yeah, it would be top.
Right.
Oh, yeah, you're right. Sorry. I'm looking down thinking left. Okay, So when we talk about leading and lagging, the leading strand, when you're using your DNA polymerase, it can just remember that I was having you model bead DNA, and you guys were facing one way and you guys were facing your back to me. So in the leading strand, you can just zip right along and bring in the free nucleotides. But on the lagging strand, because they have your back, you have to wait for some of it to be unzipped. Who does the unzipping again? Helicase. And then turn around, and it's called back stitching. Because it's like you're already passing the work you need to do, and then you're turning around, and then you're doing the work. And then you're waiting for more of it to unzip, turn around, and doing the work. These little independent fragments of DNA, what do you call those? Okazaki fragments. Now, in reality, they are 1,000 to 2,000 base pairs long. So it's not like they're tiny, okay? But those are called Okazaki fragments. We refer to DNA replication as being semi-what? Conservative. Because you conserve one old strand. Each old strand serves as a template for the new strand. Right? And let's go ahead and do a little DNA replication. Oh, let me ask you this, because I will ask you this. Okay, let me take one. If this is the leading strand right here, let me just put this away. If I was going to make DNA off this leading strand, tell me the series of bases that I would have right here.
T, A, C, C, G.
Good. If I was going to make mRNA off this strand, what would it be? Off this DNA. I don't make, yeah, off this is the leading strand right here. What would the mRNA be?
U, A, C, C, G.
Good. Every 3 bases on here would be referred to as a what? So this is a codon and that's a codon. Do you agree with that? The tRNA that's complementary, to these would be what?
A.
U. That's an anticodon, right? And the tRNA would have this anticodon and then some amino acid on it, right? Okay, what would be the next anticodon?
G, C.
Good. We all right with that? Yes.
Which ones would you refer to if you're like looking at a chart and trying to find an amino acid?
Which one do you look up? Tell me. You always look up the mRNA, the codons. And you know how you're going to remember that? What does the M stand for?
Messenger.
Yeah, you always look up the messenger. Always look up the message. Okay? Because remember, when we make that message can change depending on what we cut out, right? So I'm not going to look at the DNA because part of it might get cut out. Cut out the introns, remember, before it leaves the nucleus. So I'm not going to look at that. And also, you have several tRNAs that carry the same amino acid. So we're not going to look at that either. We're going to look at the message, and that would be the mRNA. Okay? The process by which we use this red to make the DNA, that's replication. The process by which we use this red DNA to make mRNA, what is that called? Transcription. The process of using this mRNA to ultimately build our protein, we call that translation. Good. Okay, good. I want to make sure you understood that. All right, so let's do our DNA replication. Here we go. Topoisomerase, say that word. Topoisomerase undoes the supercoiling. Then we unwind and unzip helicase. We have a leading and a lagging. We're going to use DNA polymerase 3. Boom, boom, boom. Right away, like on our leading strand. And then on our lagging strand, boom, boom, boom. What do we call these fragments? Good. What do we need to stitch those together with? Ligase. Then our newly formed DNA is going to twist up, forming sister chromatids held together by a centromere and separates during anaphase, right? Because we did the S stage, and then this would be mitosis, right? We okay? Now, the process of transcription, actually, let me show you this. So transcription, you're using the DNA to make mRNA, but in transcription, you also use the DNA to make tRNA, and you also use the DNA to make rRNA. All the RNAs come from DNA. They are all transcribed, but only the mRNA is actually translated into any kind of protein. But all of your RNAs, here are your 3 RNAs, the mRNA, tRNA, and rRNA. rRNA will go be a ribosome with some proteins. tRNA will pick up an amino acid, be transfer RNA. mRNA will just be, okay, because that's the message. So the message will go get on the ribosome, and the tRNA will bring in amino acids. Okay with that? The process of making the RNAs is transcription. The process of building a protein on the ribosome is called translation. Good. All right, and all of this wraps into what's called the central dogma. DNA making more DNA, replication. Transcription using DNA to make RNA, translation using the RNA to build a protein. Now, what we know about this is before we had all the DNA, so we just copied all the DNA because we were making a whole new cell, we needed everything. Now, we want certain parts of that DNA copied into RNA. So we're just unwrapping certain portions of it. And to know where to start, we know RNA polymerase what? Binds to the promoter. And we've now learned in our introduction to the next chapter how important the promoter is, right? Because it sits next to the operator. Remember all of that? Okay. So RNA polymerase binds the promoter, and the DNA is unzipped temporarily, and you make an mRNA copy. That mRNA copy is going to need to get modified. You're going to cut out some pieces of it. You're going to protect it by putting on a cap and a tail. And then it's going to leave the nucleus and go out and get on a ribosome to be translated. So let's make some mRNA. Let's do some transcription. So we're going to unwind and unzip, helicase. We have a leading, and we're going to put the lagging strand away. Now we're not going to use DNA polymerase. What are we going to use? RNA polymerase binds to the promoter. We're going to go boom, boom, boom. We're going to make our mRNA. We just use the leading strand. Pull off the mRNA. Set it aside. Go get your lagging strand. Zip it back up. Put it away. Now take your mRNA. Put on a cap and a tail. And you cut out the introns before it leaves the nucleus. I'm going to do better this time. DNA unwinds, unzips, helicase, leading, and put the lagging strand away. Now we're using RNA polymerase binds to the promoter. Boom, boom, boom. Make your mRNA, peel it off, set it aside. Go get your DNA, zip it back up, put it away. Now we're going to put on our cap and our tail. And you cut out the introns before it leaves the nucleus. Perfect. Now, we're going to go out, take our mRNA, go out on a workbench. We remember every 3 bases on that mRNA is called a codon. It codes for an amino acid. Who told that codon to be that codon? The DNA. Make your mRNA, right? So if the DNA said cat, then the mRNA codon would say what? C with G, A with U, T with A, right? So the codons were determined by the DNA. The DNA determines what the mRNA is going to be. The mRNA determines what tRNA is going to be on it and what amino acid it brings. So ultimately, the DNA determines the protein, right? Through a series of intermediates. Okay, so when we look at translation, the mRNA is first going to hook up with the small ribosomal subunit because there's 2 parts to it, and the large. And here's the very first codon, AUG, and here's the very first tRNA that's going to land. If the next codon was G, okay then. If the next codon was GGG, what would be the anticodon? CCC. And it would be carrying some amino acid. Could we find out what amino acid it's carrying? Yes, we could. What do we need to look up? What do we look up?
The anticodon.
We don't look up the anticodon, we look up the codon. Because the codon's on the mRNA. What does M stand for? Messenger. So if this codon is GGG, the anticodon CCC, but what I look up to find out what this is, always look at the mRNA. Yes?
Wait, so in that diagram, is the codon like the mRNA that coded for an amino acid, or is the codon?
The codon is a series of bases on the mRNA. Every 3 bases on the mRNA is a codon.
So the anticodon would be on the ribosome.
The anticodon is part of the tRNA, transfer RNA, so it knows where to land. Does that sound familiar?
Yeah, that's what it kind of.
Yeah. So I would go look up GGG. So I'm going to go look up GGG. So these are my G's, over to my G, and then my G. It's glycine. That's what that tRNA would bring. Right? So here, let's do this. Maybe this will help us. Put on a cap and a tap, and you cut out the introns before it leaves the nucleus. And it goes out and gets on a ribosome. First the small, right, then large, yeah. And every 3 bases on the mRNA is called a codon. Who determined the bases on the mRNA?
DNA.
The DNA, the leading strand of the DNA, yeah? Okay, now we're here. Every 3 bases is called a codon. It is complementary to a anticodon. So I said if this is G, what did I say on that? Did I say GGG?
Yeah.
So if this is GGG, then the tRNA landing has to be CCC. He's going to land here. What amino acid is he carrying? Glycine. So what happens when he lands, then methionine's going to come over here, and he's going to get connected right here, and you're going to have methionine, and then this tRNA is going to leave. Okay? And this ribosome is going to shift so that this guy is not in the A site, he's now in the P site, and a new guy will land in the A site on the ribosome. So it just keeps going along. New guy lands on the A site, then he gets dumped on, and the ribosome shifts. And last guy leaves. Then a new guy lands on the A site, and the guy in the P site dumps his load. Keeps dump, dumpity dump until you reach a stop codon. Okay? And that's what we're seeing right here. New guy will land. This guy will switch his over to the new guy. And that's going to keep going. New guy will land, switch over to the new guy until you reach a stop codon. Here it is, UGA. Let's go look up that stop codon. UGA, right here, stop codon. How many stop codons are there? 3. How many start codons? 1. Okay. All right, and then there you have in your book like a really good, like put it all together kind of picture in your book to use too. Yes?
When going back to the amino acid chart, like if we have a question on that on the quiz, will you give us the chart and we'll
Yes,
also
yeah, of
start
course.
it?
No, you have to memorize them all. Get started. Hurry up.
Yeah.
No, I would give you a chart. Have fun. Look this one up. All right. Questions on that? Okay. Then the next part we did, okay, is we talked about the genetic information. We said, number 1, it has to be able to store information, it has to be stable, and it has to mutate. Okay? Mutations are a normal part of life, okay? And this is a mess up somehow in the DNA. Causes of mutations, you could have just made a mistake when you were replicating. Maybe more likely that you're exposed to some mutagen in the environment, or it could be a transposon causing your mutation. What are transposons? Jumping genes. And this is evidence of jumping genes because it's removing itself and plopping itself in the middle of a gene, and it's changing and altering the color. Those are all pieces of evidence that transposons are out there. I'm not going to be able to use this one because it's going to be, yes, it's totally messed up. Okay, so if we had the DNA, the big fat cat, okay, the big fat cat. So we're just making 3-letter words because that's easier for us to understand than codons. Yes? No, this is going to be frame shift now. Frame shift. So if all of a sudden now your first codon, right, is the big fat cat, and you can understand that, right? But if for some reason this E right here got deleted, then the first codon is not THE, the first codon is going to come off as what? THB, then the next one would be what? IGF, the next one would be ATC, and then AT, yeah, okay? It doesn't make any sense this way because everything is perfectly set up. Every 3 bases is a codon. So that's why it's called a frame shift. Your whole point of reading, your frame is off by 1. So when you have a deletion of just 1 base, that's really bad. It would be better, it would make more sense if it was this. Okay? Here, I didn't do a deletion, but I put the wrong letter in there. That's better, because you can still kind of understand it, right? Big fat cat. You can still kind of understand it, that point mutation. Now, this could be really bad and cause a lot of problems, or it could cause no problems, but a frame shift always causes big problems. Okay? And then that led us to talk about, that was it. That was it. That was the end. Yay! Yes?
What's a codon if just 1 letter changes?
Okay, so if it just changes to the wrong letter, it's called, or something like that, then it would be a point mutation. But if you eliminate a letter altogether, that's a deletion. Or you add in a letter, that's an addition. Additions and deletions both cause frame shifts. Yeah, additions and deletions both cause frame shifts. Yes?
So when you're determining what the amino acid is, you look at the codon, not the anti-codon.
Always the codon.
Okay.
Always. You always look at the mRNA. Always. Well, how are we going to remember that? M stands for?
Messenger.
Always read your message. Read your messages. The codons help you read your message. Okay? Yes?
Could silent mutations eventually lead to like at first it's not a problem and then another
It may
mutation
never
happens?
be a problem. Yeah, when we looked at silent mutations, the regular DNA was supposed to read ATG, the mRNA is UAC. If you look up the amino acid, codes for tyrosine. That's normal. Here, ATA still leads to tyrosine. So that's a silent mutation. It could maybe never cause you any problem your whole life. Okay? Because when we look back at the code, right, some of these, look, it doesn't matter. AGU and AGC both code for serine. So if you get a point mutation here, no big whoop. You're still getting the same amino acid. That's why it's called a silent mutation. Look right here. As long as you get a G and a C in your mRNA, what are you going to get? Alanine. As long as you get a G and a U, what are you going to get? Valine. As long as you get a C and a U, you're going to get leucine. As long as you get a C and a C, you're going to get proline. All right? So if you get a mess up there, it's no problem. Okay? But other ones, it will make a difference. If instead of CUU, you had CCU, instead of leucine, you would get proline. That could be a problem. Right? And that was back here. Nonsense is when you got a stop codon in the middle of your protein building. You're like stopping in the middle of it. Missense is you get the wrong letter in there and it causes a problem. Okay, what else? Yes?
Would you ever have a problem with the reader or no?
The ribosome, the mRNA goes through the ribosome, and as long as your tRNA can land there, because it's only going to land where it's complementary, then you should be fine.
So it's not going to read a T as an A and then
No.
that's helpful.
I mean, there's something called the wobble hypothesis, but there's nothing in there that you need to worry about. Yeah, I mean, there's a little more to the story. I'm making it a little shorter. Okay, what else? Good. Do the practice problems within your chapter. That'll help you a lot. Okay, and remember, you don't have any additional objectives for this chapter. Right? Is it this chapter?
No, it was this
Yeah,
one.
yeah,
I checked last night.
It has it for 25.
Yeah, not for 24. You have them for 25, but not 24.
I like those questions.
Okay. They are free.
Cool, I need to
All right. Have a good evening. Make good choices.